Welcome to Microscopic Marvels. All right, let's get right into it. Today, we're tackling a real cornerstone of microbiology, something every single MBBS student needs to know inside and out. We're talking about the Ziehl Neelsen stain, which you probably also know as the acid-fast stain. This technique is absolutely essential for tracking down some of the most stubborn and important bacteria out there.
So here's what we've got on the agenda. First, we'll cover the why, the whole acid-fast principle. Then, we'll jump into the procedure, step by step. After that, we'll figure out how to interpret the results, see why it's so important in the clinic, and wrap up with some key modifications that are super high yield for your exams. Sound good?
All right, let's start at the very beginning. You might be wondering, why do we even need a special stain for these particular bugs? I mean, what's wrong with the good old Gram stain? Well, it all boils down to this really unique property they have called acid fastness.
So, acid fastness. It's pretty much exactly what it sounds like. It's this incredible, stubborn ability of certain bacteria to just grab onto a dye and not let go. No matter what, you can douse them in strong acid and they just hold on tight. It's kind of like their bacterial superpower for real. And the secret to this superpower? It's actually pretty cool. These bacteria have a cell wall that's just packed with this waxy stuff called mycolic acid. Think of it like a thick, waterproof raincoat. It makes their cell wall almost impossible for normal stains to penetrate. Now, a little history for you. Paul Ehrlich was the first to notice this, but it was actually Ziehl and Neelsen who came along and perfected the staining technique that we're still using today.
Okay, so we've got the why down. Now, let's head over to the lab, metaphorically speaking, and look at the how. We're going to focus on the classic procedure here, the one known as the hot method.
The whole thing really just boils down to three main steps. Step 1, we force the main stain into everything. Step 2, we use acid to wash that stain out of, well, almost everything. And step 3, we add a different color stain, a counter stain, to make the background pop for contrast. See? Simple, right? Let's get into the nitty-gritty. So first up is our primary stain. We're going to flood the slide with this really potent reddish-pink dye called Carbol Fuchsin. But here's the most important part of the whole process. We have to gently heat the slide. You want to heat it just enough to see vapors rising. Why? Well, that heat is what melts that waxy raincoat of mycolic acid, forcing it to open up so the Carbol Fuchsin can get way down deep inside the cell wall. Just a quick tip, don't let it boil and definitely don't let it dry out. Okay, next is the step that separates the stars from the background actors, decolorization. For this, we use a pretty powerful 25% sulfuric acid solution. This acid is strong. It's going to strip that red dye from everything. I'm talking human cells, other bacteria, all the background gunk, everything, except those stubborn acid-fast bacteria. They've locked that stain in and their waxy coat is protecting them. When you've done this step right, the slide will look like a very light pink.
And now for the final touch, the counter stain. We just add some methylene blue for about 30 seconds. Its job is super simple. It just stains everything that the acid just washed clean. And that is what gives us that beautiful contrast we need to see what's what under the microscope. It sets the stage for the big reveal.
Okay, listen up, because this next part is pure gold for your exams. I know, trying to remember all these steps and chemicals can be a real headache, but don't worry, I've got a killer mnemonic for you that's going to make it ridiculously easy.
All you have to remember is this simple phrase. Hot cars are blue. Let's break it down. H is for the hot method. C, cars, is for Carbol Fuchsin, our primary stain. A, R, is for acid, the decolorizer. And B, blue, is for methylene blue, the counter stain. Seriously, lock that phrase in your brain and you'll never mess up the order in your practicals. Ever.
All right, we've done the hard work. We've stained our slide. Now for the moment of truth. Let's learn how to actually interpret what we're seeing under the microscope, because this is where it all comes together. This is where the diagnosis happens.
So what are you actually looking for? Well, the stars of the show, the Acid-Fast Bacilli, or AFB, they're going to be a bright, unmistakable red or pink. And why? You guessed it, they're the ones that stubbornly held onto that Carbol Fuchsin. You'll see them as these long, slender rods. Sometimes they even look a little beaded. And everything else? The background, the other cells, any other bacteria? It's all going to be a cool blue because it took up the methylene blue counterstain.
So, okay, great. We see red rods on a blue background. But what does that actually mean for a patient? Let's shift gears now and talk about the clinical side of things, how this simple stain becomes a life-saving diagnostic tool used every single day.
Okay, lean in a little closer for this one because this is a classic exam topic. When we're hunting for Mycobacterium tuberculosis in a patient's sputum sample, it's not good enough to just say, yep, it's positive. We have to actually grade the smear. We have to quantify it to tell the clinicians how severe the infection really is.
So how do we do that? We use specific guidelines to quantify what we see. For example, if you scan 100 different fields under the microscope and see absolutely nothing, that's negative. But if you find just 1 to 9 of those little red rods in that entire scan, we call that scanty positive. And then you have the other end of the scale. If you're seeing more than 10 bacilli in every single field you look at, well, that's a heavy load, a 3+ positive. This whole system is crucial for giving doctors a really clear picture of the patient's bacterial load.
And don't think the ZN stain is just a one-trick pony for TB. Not at all. It's also absolutely critical for diagnosing leprosy using skin smears, and for identifying other bacteria that are what we call partially acid-fast, like nocardia. Heck, we even use it in parasitology to spot oocysts like Cryptosporidium in stool samples. It is an incredibly versatile, powerful workhorse in the lab.
All right, for our last section, let's quickly go over a few important modifications. It's good to know that the classic hot method isn't the only game in town. Knowing the variations is super important, both for real-world lab work and, of course, for your exams.
Okay, here's the massive tip. Put a star next to this in your notes. This is a Viva and MCQ favorite. Remember that 25% sulfuric acid we talked about? Well, it's not a one-size-fits-all deal. You actually have to change the strength of the acid depending on which specific bug you're trying to find.
Just take a look at this. M. tuberculosis. It has that super tough, waxy coat, right? So you need a really strong 25% acid to even try and decolorize it. But then you have M. leprae, the bug that causes leprosy. It's less acid-fast. Its raincoat is a bit thinner, so we only need a 5% acid. And for even weaker guys like nocardia or those parasitic oocysts, we dial it way down to just 1% or even less. Knowing these different concentrations, trust me, it's a classic exam question.
And just to round things out, you should know about a couple other key variations. There's something called the Quignon method, which is basically the cold method. It gets around the whole heating step by just using more concentrated chemicals. You might also see different reagents used sometimes, like acid alcohol as the decolorizer, or even malachite green as the counterstain instead of blue. They're all just different tools in the toolbox to get to the same result.
Which kind of brings us full circle to a final thought. How is it possible that one single staining technique can be so versatile? How can it help us diagnose everything from tuberculosis to parasitic infections? Well, the answer lies exactly where we started, in that unique, waxy mycolic acid cell wall. It's a simple but absolutely brilliant evolutionary feature that's shared by more than one microscopic marvel.
If you like this video, then please like, comment, and subscribe.